We are happy to welcome you at the online lecture series from the NGS-CN Summer School 2024. As most of you already know, NGS-CN stands for the Next Generation Sequencing Competence Network. Our network has started as a DFG-funded national collaborative initiative in 2019, and up to 2023, there have been several annual calls for projects to be sequenced at our core genomic facilities across Germany. From 2024 on, you have a new exciting opportunity to co-apply with one of our genomic facilities for a high-cost sequencing project, which can cost up to 1 million euros. If you ask me, it's worth to check it out. Go for it. We leave the link in the footnotes, and here is a QR code for your fast access. Welcome aboard, find a comfortable seat, get a drink, and let's listen to our next speaker, shall we?
I'm Maximilian Krause. I'm at the Dresden-concept Genome Center, one of the four NGS-CN facilities that Julia just mentioned. And today I want to really set the foundation for your understanding for the next 2 days. And with foundation, I really mean, let's say, a quick brush overview about what technologies are out there and the technological nitty-gritty details so that you are best trained to make a good choice for your NGS approach and which technology to choose. Julia already mentioned, we are a network of sequencing centers in Germany. There's 4 main sites, 4 genome centers, from which the West German Genome Center is separated over different locations. I'm at the Dresden-concept Genome Center, so at the east of Germany. And this is where I'm sitting right now with the gray wall in my back. I'm really happy to see more than 100 participants, so I tried to envision you all in front of me instead of the gray room in which I'm sitting and the gray weather outside, but I'm actually happy about the gray weather. So with no further ado, I try to really guide you into sequencing technologies. My talk will be split into 4 main parts. The end is really just a slide, one slide. I will quick brush you over what DNA in the genome actually is, so that we all speak the same language. I will then talk about what sequencing is and where it comes from, so a little bit of history there. And then we will talk about short read and long read sequencing in specific, because these are like the technical details that matter for you. So with no further ado, let's start. So the DNA, I think everyone in this room should know that the DNA is a biological molecule that is made of deoxynucleotides, so small building blocks that build a biopolymer that then forms a double helical structure by non-cohesive binding from the two reverse complement strands. And the building blocks are adenine, thymine, cytosine, and guanine that sit on a phosphate backbone that builds the single strands of the DNA, right? This is really very basic. You can talk, I don't know, 2-3 hours about DNA by itself. But for today's talk, this should be enough. Why it is so important is that the DNA actually encodes all the information that is needed to build life forms as we know them, whether it's humans, whether it's animals, whether it's plants, bacteria. The actual building blocks are encoded in kind of a blueprint, and that is stored in the DNA in a double helical structure. And that is why it's so important to understand DNA, because with knowledge over DNA or probing DNA, you can understand and probe biological life. DNA is actually encoding the biological life, and that comes in gene blocks, right? I think the name gene, everyone should know that's sitting here. But the idea is that DNA is transcribed into RNA, RNA is transcribed into proteins, and the proteins are the building functional units of cells, amongst other things, right? So this is very basic, but it's a basic principle of biology, the transmission of information from the stored form of DNA towards proteins that then restore function. However, the genes are the pieces of DNA that encode RNA and proteins. There's more to DNA than just genes, right? Actually, only 1% of the DNA in eukaryotes encodes RNA. We talk about 20,000 genes and roughly like 30 to 40 million bases in a 3 gigabase genome that are actually only encoding the genes. Other DNA elements are regulatory elements like enhancers and promoters that regulate when genes are transcribed, how they are transcribed, timely transcription and amount of transcription, by folding onto each other and influencing the transcription process. I again stop here. There's much more to learn about this, right? I don't know a whole lectures series over this alone, but for today, that should be. Also quite important, and that will influence also the talks that we hear later. DNA is not just there as a single-stranded or double-stranded molecule. DNA is actually organized in chromatin by higher organization. And this is done by histone proteins around which the DNA is wrapped. And these histone complexes, they can form these on a string model that then gets folded on top of each other, building the chromosomes that we know, right? The spatial orientation, the spatial interactions, and also the chromatin organization, whether it's compact or loose, influences the gene expression independent of the actual machinery running there, right? So it blocks the machinery from binding, it can block transcription factors or other factors to bind and enhance or reduce expression. So this is not just happening on a beats on a single strand. It is highly complex, highly organized. And important to know as well, the regulation of transcription can be encoded in the DNA, but also on top of the DNA. And with that, I mean actually factors that influence the compactness or the binding affinity to DNA with these histones. Again, you can talk about much more about this, right, for lectures. For today, it should be enough. But I hope with this, I can give you a rough sketch of where DNA is sitting, and that all influences how you can probe it and how you can analyze it later. So in later talks, you might hear about parts of these. So let's go to what is sequencing. These next sentences are written by me. I don't see myself as the wisdom itself, right? You will find much better sentences most likely online. But I try to nail it in a way that it helps my talk. And sequencing for me describes the process of deciphering a repeated structure and depicting it in an unranged linear chain of symbols. So quite important to say this in this way because sequencing is not limited to DNA. It's not limited to biological molecule. You can sequence much other things, right? So it is a process of deciphering a repeated structure and depicting in symbols so that you can read it without actually having the thing in your hand. And for biology, it is the process of characterizing the primary structure of biopolymers, for example, DNA in this case, and depicting it with alphabetic characters symbolizing the building blocks. And this way, we can actually decipher and read and store the information independent of the actual biological knowledge, right? So that is sequencing for me. I like to also depict sequencing as akin to storing knowledge in humankind, right? I mean, we as a human species, we became, let's say, quite dominant on the world because we were able to store information, share it with our next generation, and by this build information and knowledge much more over than just what you experience in your own lifetime. And this is done by books. And the revolution in terms of sharing knowledge happened with the book printing, where you can actually depict what you have in knowledge in writing and then print it and manifold it so that you can share it amongst many other things. And with sequencing, this is kind of the similar. Not everyone can probe DNA, but everyone can read DNA if it's stored in a readable format. And going from DNA in nature to a readable format, this needs sequencers as books need book printing, right? And this is what we talked about. So short history of sequencing. For most people, the story of history of sequencing and DNA starts with this picture, which is Watson and Crick showing the double helical structure of DNA, but it actually started much earlier. Already in 1880s, Friedrich Micha, a Swiss scientist and chemist more, has proposed that it is actually an acid, an acidic form of biological molecules that is in every life that is most likely storing the information. He was not saying more as the chemistry that sits there, but he couldn't decipher it and read the information out. But just showing that the knowledge is always in science, right? Building on top of each other. And also not to forget picture of the double-stranded helix as it was built by Watson Crick is also building up on X-ray structures that are done by, now I forgot the name, damn it. Julia, Daniela, can you help me? Why is it gone?
Rosalind Franklin is what you're looking for?
Yeah, thanks. Rosalind Franklin. Just, you know, I put the picture there to say it's not only a male story, right? And non-protected name, sorry. Exactly. So this, she was great in making the X-ray pictures that actually are from nucleic acid, and based on these pictures, actually could then find out that it's double helical structure. Sequencing started really in the 1980s to 1990s by Sanger sequencing as a process in which you copy the DNA, and whenever you build in a base with a specific chance, you build in a fluorescent molecule. And when you then perform a gel electrophoresis to separate DNA based on its length, you get a ladder picture with colors. Nowadays, colors. Earlier, it was 4 different reactions that say which base is there with the highest security. So this shows a depiction of how Sanger sequencing works, right? The longer the molecule, one by one step, you can decipher A, T, G, and so on and so forth. In real life, it looked like this. Picture this to decode it by hand, right? You could also use computers, but it's not very easy and not very straightforward, right? But this is how the human genome was started to be deciphered in the early 1990s. And machines looked like this at that time, right? So this is actually a picture of the Sanger Institute in which the human genome was sequenced in the sequencing was started. And all these are Sanger sequences with their own compute units, and they would all analyze these kind of pictures. We came a long way since then. And a huge boost happened with the idea of hierarchical shotgun sequencing. And the idea there is that you hack the DNA into pieces, create short pieces, and then sequence all these short pieces, millions and millions of those, so that you can actually bioinformatically backtrack the DNA information by putting in millions of short pieces with high compute power on top of each other, stitch them together so that millions of short pieces make hundreds of big pieces and hundreds of big pieces make one big genome. So this is actually really a concept that then was brought into life with molecular biology technologies. And this is what next generation sequencing stands for. Next generation sequencing, or NGS, is the idea of massively parallelized sequencing of very short fragments, which was nowadays dominated by Illumina machines. So everyone sets NGS sequencing akin to very short fragments and very short fragment sequencing to akin to Illumina, even though this is not the whole story. But for today, this should be enough, right? Also to depict this, we live in a golden era of genomics because we don't think about price anymore when we do sequencing. We do, but it doesn't play a big role. Depicting this, that in around 2001, so not even 25 years ago, the human genome was finally solved. And this costed more than $100 million, I think always almost up to $1 billion. And sequencing at that time, a whole human genome was around $100 million. That was the price tag. Yeah. And then with technical evolution, you, as always, by being more efficient, using better technology, you can reduce the cost. And this orange line shows the normal way of, let's say, industry improvement without big breakthroughs. And you see that sequencing became cheaper, but it would still cost 10 million euros in around 2007. Funnily enough, this is when I started my molecular biology studies. Back then, 10 million euros or dollars would it cost to sequence a human genome. But then really big improvements made the price drop. And when I finished my studies, it would be already 2 orders of magnitude cheaper to sequence a human genome. And it became affordable to actually use sequencing not only for deciphering the genome, but with much more different applications. It stalled quite a while on this level, but nowadays we go towards the $1,000 genome. And I think as of today, we are even below $1,000 per genome, or we can be if we want to. It's super efficient, right? So we are really in a golden age for sequencing technologies because we don't have to worry about pricing anymore. We can really choose the best technology. Okay, to summarize, DNA is a biopolymer. I guess you all know this. DNA encodes the blueprints of proteins and with that, the function of life. And this is not sitting around simple, it is compacted into chromosomes. And sequencing for me describes the process of identifying the DNA content and depicting it as alphabetic characters. It is earlier done with Sanger sequencing by chain termination, and shotgun sequencing revolutionized the process, leading to next generation sequencing. Okay, and the next generation sequencing is actually usually sequencing by synthesis. With this, let's directly jump into short-read sequencing. As I said before, most people say when they talk about short-read sequencing, they talk about Illumina. And this comes because Illumina was the company that dominates sequencing over the last decade or so. They were very good in the beginning in scientific technology improvement, but also rapid marketing and then taking over other companies so that they became basically a quasi-monopolist. There's other technologies out there. I will mention them later, but short-read sequencing and sequencing in general is dominating. Oxford Nanopore and PacBio, I will mention them later as well. These are the long-read technologies. And you see that they take a share of this, and I guess they will grow. But as of today, Illumina is still the big boss in sequencing. When we talk about short-read sequencing, I mentioned already, it's millions parallel site sequencing. The usual process is that you start with DNA, you cut it into short fragments because this is what you can sequence. You create a library. Library construction actually means that you functionalize the ends so that you can work with them and you know them. This is what library construction stands for in a very short term. And this is true for all technologies. Using these adapters, you can immobilize the DNA on a surface and then sequence it, creating millions and millions of shortcuts. This is the basic concept. For Illumina, this works in a way that you flush in single-stranded DNA molecules. You have a surface that is labeled with DNA oligos that fit to the, as I talked, adapters that you bring to the end. The flushed in molecules will be captured, and by bridge amplification, they will be amplified on the spot, creating exact copies of the same DNA molecule on the spot, 40 to 50 of them. And 40 to 50 of them at the same spot will then create one cluster, and one cluster stands for one DNA molecule that will sequence. On these flow cells, you can have millions, if not billions, of those clusters, either predefined by having spots of those oligos, or they're just by sparse labeling there, you can spot them randomly. Okay, the sequencing happens by sequencing by synthesis with reversible terminator chemistry, and you sequence millions and millions of molecules base by base. So one cycle is one base, and you do all 1 million or billion spots at the same time. And the cycle speed defines the sequencing speed. So this is how it works. You flush in a primer and the nucleotides. The nucleotides are built in. You then image, you then bleach, cut away this, wash out everything that's left, and bring in the next cycle of nucleotides. They will build again. You can make again a picture, you bleach it, decross-link, and then you start the cycle. Yeah. And what you get is pictures where one spot is all 30 to 40 molecules at the same spot, lighting up with a different color, and the different color stands for it. Just to mention, this picture is outdated. I like it because here every base has a different color. Nowadays, it's not that every base has a different color, because otherwise you would have to take 4 different pictures. Most sequencers work with either 2 colors only, or there's some that work with, I think, even only 1 color, and then it's encoded by whether it's on 2 colors is combinatorial, or just 1 color, or 1 color, or no color, right? So, but for depiction, this is the picture. In the end, what is most important is that you make a copy locally fixed on a flow surface, and then your sequencing process is actually imaging-based process, right? This is it in a nutshell. Illumina has a big range of machines, starting from mini-seq or MiSeq that only can make several 100,000 to 1 million or 10 million reads. And then each machine, when they become bigger, they can produce more reads at the same time, leading up to NovaSeq and even the NovaSeq X Plus that can run 2 flow cells, and each flow cell can run 25 billion reads. So massive amounts of sequencing in there, right? As I mentioned, there is alternatives, especially nowadays. They become more and more prominent. The two spearheads that fight Illumina basically on their own market, which is MGI Technologies and Element Bio. Just a brief recap, both of them are also fluorescent labeling and imaging, but the library prep is a little bit different. So for MGI Technologies, you create a circular molecule that has the insert of your interest in there. You then amplify it in the tube, creating so-called nanoballs, DNA nanoballs, DNA. This is special for MGI. These nanoballs contain copies over copies of the actual molecule. And then you flush in these nanoballs onto the surface of the flow cell. You bind them on spots. And then sequencing happens again, cycle by cycle by cycle, base by base. And here it is not double-stranded molecules that sit on the surface, but this nanoball, and then the nanoball lights up whenever you have one base sequence. Element Bio is a little bit different, but actually a mix kind of for me of MGI and Illumina. Here you have single-stranded DNA that captures this on a circle on the flow cell. And then instead of flushing in DNA nanoballs, you flush in these circular DNA molecules. And then you amplify the circle on the spot, creating one long spaghetti of DNA. And the sequencing happens not by sequencing by synthesis, adding nucleotides. Instead, you flush in a polymer that has fluorescent molecules sitting there, and on long chemical arms, the actual bases sit there. So that you immobilize just by binding, not by synthesis, but by binding the fluorescently tagged biopolymers on your string. And then depending on what binds, you get one color. This apparently gives higher quality because you're not dependent on single fluorescent nucleotides sitting there, but one blob of fluorescent binding by multiple arms. And by this, you can actually increase the efficiency of binding. Again, these technologies right now don't have a big market share, but they are building up on Illumina technology, so you can almost one-to-one replace them, both in data that comes out of the machine, and also in what preparations you have to do in advance. It is only the way how the sequencing works is slightly different. This is how I would describe it. Okay, summarizing short-read sequencing. I say in bracket Illumina short-read sequencing because it's almost true for all short-read sequencing is based on sequencing by synthesis, Element Bio sequencing by binding. The readout is always for all technologies fluorescent signal that is per single base on millions of clusters at the same time. And the output are millions, if not billions, of short reads in the length of 100 to 250 or maximum 300 base pairs. The advantages of these technologies is that they're, especially Illumina, is very established. You know exactly usually what you get because there's a big knowledge build up already in the past years. And a lot of tools are built up on exactly this data type that Illumina is producing. The data quality is super high. We talk about 99.75 accuracy per base, and you get millions and millions of reads, so you can do good statistics, right? And it is rather affordable because you can parallelize massively, and optimization has happened. The disadvantage is that the machines cost a lot of money also in maintenance. So you first have to have a blob of money that you can buy the device and buy yourself into the technology. Short reads can be a disadvantage. And the sequencing by synthesis actually means that you have to wait all the way to the end of sequencing to get the read as a whole, right? So let's switch a little bit gears to long read sequencing. I like to start with this picture because end of 2022, long read sequencing, both technologies were mentioned as method of the year. And since then, they really picked up in speed and became more common nowadays. There's two big players, which is PacBio or Pacific Biosciences. They recently changed their logo to be a little bit more bright. And Oxford Nanopore Technologies is the team. Let's start with PacBio. PacBio libraries are not single-stranded or double-stranded DNA molecules. They are, but the library itself that is not double-stranded, but it is a bell-shaped adapter on both ends. So this is very special to this. It's called Smart Bell Library. This library gets loaded onto a flow cell that has nano wells, so super small diameter wells, basically only allowing one DNA molecule at a time to sit in there. And then the bell here is bound by a polymerase that is immobilized at the bottom, not floating around, but immobilized at the bottom. So basically this enzyme catches the DNA and then it sits there and holds the DNA in place, blocking this well for other molecules. Sequencing is happening kind of similar to Illumina. This is why I also start with this long read technology, because you again have an enzyme that brings in nucleotides, and when the nucleotides are built in, fluorescence is detected, and then it is built. But instead, different to Illumina, there's two main differences, and one is that you don't have cycles where you bind, image, block, make the next round, but you have a constant turnover. And this creates not just one image per cycle, but you get actually fluorescent light pulses over time. And why do you get so sharp light pulses is the second difference. The imaging is not a plain picture of the whole slide. Instead, you actually have a laser that comes with an angle from below in a specific angle. And because of the specific angle, it only lights up a super small area at the bottom of the flow cell, not the whole nanowell, but really only a super small area at the surface. Most of the laser is deflected and goes away, and it doesn't go all the way through. So you have less photo bleaching, and you only detect the fluorescence if it's really sitting in the enzyme. And this is kind of depicted here. You don't see any fluorescence if it's floating around here. You only see it if it's here. And then the building in releases the fluorophore, it floats away and is not lighting up anymore. And this way, you can make live sequencing readout base by base again by only that what is bound you sequence. But it is a continuous process, not a cycle flushed process. And you have less photo bleaching because you only activate and observe what is really sick. Long talking for a very simple process. In physics, it's called TIRF microscopy, total internal reflection fluorescence microscopy. If you want to Google more, this is the term you have to search for. What PacBio is delivering is single molecule sequencing, as opposed to Illumina or MGI Element Bio, where you have copies of the same molecules sitting all together. Here you just have a single molecule, you observe only a single molecule, and the technical limit is at 100 to 150,000 bases per read. This is when this enzyme finally gives up and you have to kind of replenish it. PacBio is specifically focusing on 10 to 15,000 base per fragment length for high quality. And this is happening by making a circle and then sequencing the same circle over and over and over again. And if you look at the numbers, you sequence each position roughly 10 times, and then you can superimpose them on top of each other, learn and polish out mistakes. And by this, you get a much higher quality if you sequence every molecule 10 times, as compared when you go to the maximum technical limit and sequence each base only once. Yeah. So the 10 to 15 kilobase fragment length, which is called high fidelity sequencing, creates a much higher per base accuracy. How it works is a nanostructured flow cell with an immobilized Phi 29 polymerase, and it sequences roughly at the speed of 5 bases per second. And then the fluorophore is retained while it is sequenced. Okay, the sequences look like this. This is a small sequel2. I say small. Yeah, you see how big it is. It creates roughly 2 to 5 million reads. and 20 to 30 or up to 40 gigabases of information per flow cell. You can load 4 flow cells at once, but only sequence 1 at. And then the newer, bigger devices behind here, it doesn't look much bigger. It is a little bit deeper. This is why there's not so big a difference here. This can sequence up to 10 million reads on flow cell, producing 30 to 100 gigabases of information officially. Mostly 50 to 60, I guess. Okay. This is now for PacBio sequencing. Again, summarized, it is based on sequencing by synthesis, read of fluorescence, and you produce millions, up to 10 million per flow cell, of long reads, 10 to 15 kilobases if you want to have high quality, 100 to 150 kilobases maximum. The high accuracy is actually better than Illumina, 99.9 versus 99.7. Doesn't sound like a big difference, but it means 2 bases in 1,000. 2 bases are wrong in 1,000 bases of information. And a gene is usually like the coding region of a gene is usually between 1 and 2 KB. So you have already quite some mistakes there. The disadvantage is super high initial investment costs. You need very good sample quality. So you have to work on your sample very much before you actually get there. And you need a lot. And to bring it into sequencing also needs a lot of work. And the experiment itself is rather expensive at low data quantity, not quality, but quantity. Yeah? Let's go to Oxford Nanopore sequencing. Oxford Nanopore sequencing is totally different. So we have to go away from what you remember from before. Here we talk about a membrane, a biolipid membrane, in which a pore sits, like a protein pore that has a hole in the middle. On top of this, you load your library. And in this case, the library is a double-stranded DNA with a fork-shaped adapter. And on the single-stranded fork-shaped adapter, a motor protein sits. This motor protein interacts and binds to the pore and then feeds the DNA through the pore. While DNA is stuck in the nanopore, you have less ions floating through the membrane, and this changes the electronic potential you can observe over the membrane. So no fluorescence, you talk about electronic potential over membrane. No flushing in and building and copying the DNA. It is putting the DNA with the motor protein through the pore. And basically, it is really a fundamentally different principle of observing DNA. And you decode the nucleotide bases based on the electrical potential of blocking the pore in the middle of the constriction. This is how it looks in animation. I really like to look at these animations because they are very nicely made, but we have a membrane again, the pore, the DNA ratchets through, changes the electrical current, and based on the electrical current, you can decipher the DNA. Okay, this is depicted here again. In sequencer, the smallest sequencer is actually a handheld device. It is as big as a little bit bigger than headphone box or something. Yeah, and you could copy it into USB, into a computer, and depending on how powerful your computer is, you can actually observe the sequencing in real time. So compared to what you've seen before, much smaller devices because you don't need a huge microscope and you don't need any chemical flushing that needs pumps in these kind of things, right? You load it once, it goes through the pore, and then you sequence it with electrical currents. And this is why the box is so small. A summary of how it works, right? Membrane separate 2 ion buffers. Pore allows ion flow, but can feed DNA through the membrane. The motor enzyme is actually making a constriction and a controlled movement. And 5 old pores was 5 bases sit within the pore. New pores, there's actually 12 bases sitting in the middle of the pore and influencing then the current. And you record the modulation if we are in a current, which is base specific. And you can even, and that is very specific to this technology, you can detect modified bases directly based on the electrical current signal. I want to mention here that also PacBio can sequence methylated bases. And this is based on that I mentioned before that you have a fluorescent pulse. When the base is built in, if the base that is tried to be copied is methylated, it takes the enzyme longer to build in the reverse complement molecule. And based on the time dynamics, you can actually say this base was modified. With Oxford Nanopore, you measure actually the DNA without copying it as it goes through a pore. Okay, so the Oxford Nanopore sequences look different than the others. You have super small devices. This is the smallest one. So this is the smallest sequencer. It feeds this flow cell, but you can actually use an adapter to feed an even smaller flow cell on there. The small flow cell is as big as your thumb, roughly. It brings you 100,000 to 500,000 reads, for roughly 1 gigabase of information, or a little bit less. The bigger flow cells here, as I said, roughly like this, 5 to 10 million reads, up to 10 gigabase of information. They go into this flow cell, this sequencer. The sequencer can also be built in with the screen so that you can analyze it at the same time and visualize it at the same time. Or you can have a grid ion where you can put 5 of those flow cells next to each other. The bigger flow cells, these are these. They have a much higher number of pores sitting there. They go into the big sequencers, but even the biggest sequencers is more a box of a printer size, roughly, and it's still a benchtop machine. So you can here load up to 48 of those flow cells. In the bigger flow cells, you get 50 to 150 gigabase of information. Officially, the company says up to 500 gigabase of information, but I did not hear that anybody reached that yet. And you get multiple 100 million reads out of this flow cell. Okay, summarizing this part again. Oxford Nanopore is based on ion flow interruptions, so totally different of the rest. The readout is current, that change over time. The outputs are millions of very long reads. Basically, there's no technical limit. You can sequence reads as long as you provide them. So actually nowadays it's a customer that creates a problem because too high pipetting already breaks your DNA into pieces. If you are very careful, you can reach up to 2 million bases and more. This was recorded in real life. But regular, you get easily 100 to 300,000 bases at once, and 1 million is also not rare. A very big advantage in my eyes as a molecular biologist is that you sequence the native molecule. So you don't create a copy, you don't read the copy, you read actually the molecule that you load into the sequencer. And if you didn't copy it yourself, you read what is in the cell or in the organism that you're studying. And that allows not only modification detection, but it also allows RNA sequencing for the first time. Something I didn't mention before, but any other sequencer that we talk are DNA sequences. And if you study RNA, you first have to transcribe the RNA into cDNA, then copy the cDNA, and then this is what you sequence, right? So it's a very indirect measurement of RNA. With Oxford Nanopore, you can sequence the RNA as it comes out of the cell without any modifications. And this allows to study the RNA as it is in biology. And if you have heard a lecture about RNA sequencing, or about RNA itself, RNA has many, many more base modifications as DNA. So now we are in an era where we can actually probe them. Another big advantage for Oxford Nanopore is live sequencing. We talked about that Illumina depends on the cycle time. Cycle times are 1 to 5 minutes, but you only get all reads at the same time at the end. PacBio has a speed of 5 seconds per base, and you can track them live, but 5 seconds per base is also rather slow. Oxford Nanopore can do up to 400 bases per second. And this allows you to actually sequence live and observe and collect data live. So within 5 to 10 minutes, you already have 1,000 or 10,000 reads. If you have captured something specifically before, within 10,000 reads, you already can start doing statistics and learn something about the sequence, about your result, about your sample, within 10 to 15 minutes. However, that comes with disadvantages compared to all other technologies. The quality, the per base quality is rather low. Nowadays, we speak about 97 to 99% accuracy for DNA. For RNA, it's around 90%. And that's a modal average. So you have some reads that even have lower base accuracy. And the data quantity, especially compared to Illumina, is also low. The other disadvantages, which you could also say is an advantage, but it is continuously evolving. The chemistry and the flow cell designs change rapidly recently, so that it's really hard to build up a routine and a structure for yourself, following always the same protocol because they get modulated over and over again. So it's really hard to streamline and get used to this technology. Also, another disadvantage is you cannot really plan with the output because it depends very much on the sample, on yourself, on how you create the libraries, and also on the manufacturing on the flow cell. So it's really hard to plan with what you get out in the end. And as with PacBio, you need super high sample requirements, meaning quantity and quality, so that really nice data comes out. And it is rather costly. It is comparable to PacBio, but it's much more expensive than the Illumina sequence, at least per base. With this, I'm at the end. And there's also optical mapping. I've seen that some people are looking for optical mapping. I did not cover it here because it's not really a base pair sequencer. It's really more in lighting up of sequences and therefore an indirect measurement of DNA. So this is a different concept, and I don't have it in these slides here. Summarizing short read and long read sequencing, PacBio and Oxford Nanopore, you see it here. Short read sequencing and PacBio are sequencing by synthesis via fluorescent imaging. Oxford Nanopore is DNA through a motor protein, and you measure electrical signal. Read lengths are 100 to 150 or up to 300 bases for short read sequencing, but billions of reads. Here you get 10 to 15,000 or up to 150 KB for PacBio with 2 to 10 million reads. And you have unlimited length for Oxford Nanopore, depending on your sample, and you get up to 100 million. The accuracy, the highest accuracy is with the long read PacBio hi-fi sequencing, 99.9%. You get less with Illumina, and you have less at the moment with Oxford Nanopore. They're working on technology where you get towards 99.9, but that reduces the amount of length and the amount of reads that you get. So it's not really comparable. The initial costs are cheapest for Oxford Nanopore for the small sequencer because it's just a small device, right? And it's easy. Illumina is expensive. PacBio is also very expensive. And here are the pros and cons that I listed before. For your, I don't go over them point by point. With this, I just want to mention that we talked about sequencing technologies. And for me, technologies are the tools like this drill, right? With drilling, you can do a lot. You can either drive screws into a wall, or you drill holes into a wall. It can drill. But even there's modifications. If you want to go in small corners, you can get super small, super compact ones, but they're not as powerful. Or you have super powerful ones if you really want to drill big. All of them are the same machine, but depending on what you need, you can choose the right tool. Sequencing for me is a multi-tool. A multi-tool is actually something the device itself can only do one thing. In this case, the multi-tool, it can vibrate, right? But what you can do with vibration is massive possibilities. You can either polish something, you can chainsaw something, you can drill holes, you can cut away things, right? And the technology, that is the device. What you do with it depends on you, and that is the sequencing application. So the next talks of our summer school will focus more on applications. So they will always use the same machine, but they will be used for different cases. And right now, I hope you got an overview of what kind of technologies there are so that you can choose the right tool. But what you do with this is depending on your biological question. With this, I'm at the end, and I'm happy to take questions.